the questionnaires. Mitosis. Mitosis is a part of cell divisions. We can see here types of mitosis, anestral mitotic divisions. Anestral, in this case, sometimes it will be happen in the plants. Most of the cases, anestral means there is no such astral formation, because as we know, the astral formation is not visible. Only tubulin will be take place to make the spindle apparatus. So this is type of plant mitotic division is called anestral mitotic division. Amphiastral means both sides. In case of animals, the centriole goes to the both poles and release particular astral rays. So that's why it is called amphiastral mitotic division. Intranuclear mitotic divisions means within the nucleus, the nuclear membrane does not dissolve at all. First one, nuclear membrane will not dissolve. Okay, dissociates nahi, no dissociation. And another one is spindle fibers will get separated. Spindle fibers is getting separated to each other. This is the specificity for extranuclear mitotic divisions. Multiplications. Mitotic divisions. Endomitosis. Endomitosis is very, very important, where we can see that spindle fibers basically duplicated, but there is no chance of separation of nuclear materials. So that means a single cell can contain a number of duplicated chromosomes. So if suppose when cell division has been totally completed, in that case the chromosome number will be duplicated, but the cells contain the same. The number of, if the cells initially is 1, after cell division one cell may be without any nuclear materials, another cells contain duplicated nuclear materials, because duplicated. This is basically occurred in the plants, and in case of plants it is called polyploidy. Number of chromosomes will be increased in the duplicated, triplicated, tetra numbers of increase, and it is very, very useful for crop productions. But in case of animals, it is chromosomal abnormalities has been found. Denomitosis. In case of dinoflagellates, the nuclear materials is dissolved. Sorry, the nuclear materials, nuclear membranes will be dissolved, and spindle fibers does not separate it. This is the opposites of extranuclear mitotic divisions. So discovery of Fleming's, discovery two different scientists are there. Fleming's in animals, Strasburger in plants. How you can remember? F word letter is related to A, nearest to A, and Strasburger related to P, nearest to P. So in this way you can easily remember who scientist related to which particular plant cell division discovery. An exception, mitosis is a general germinal cells, as I told earlier. This is the exceptional stages of mitosis, and we can see here. First one is, we can see early prophase. Early prophase, we can see. In this case, for you easily remembering, dissociation, association, and 3 different verbs I have used. So you just have to remember the verbs and which particular materials is related to these particular verbs. You have to just remember dissociation, association, initiation. From the earlier prophase, you can see here what is dissociated. The nuclear membrane, this nuclear membrane getting dissociated, and all the central part of the cell organelles, the membrane bound cell organelles will be dissociated. Association, dissociation, association means the chromatin fibers, it is not visible actually in the early phase, but in the early prophase the chromatin materials become visible to make chromatids. That means they are associated to make chromatid like structures. And initiation, it is related to, initiation means initiation of particular spindle fibers. Spindle fibers, it is not actually visible here, but the spindle fibers start to initiate because the centrioles goes to opposite poles. Hmm. So, sorry. Next one, late prophase. We can say here disappears. The membranes, nuclear membranes are totally disappears. Here, attachment. Attachment means the particular spindle fibers will be attached to the particular chromosomes at a particular location. This is the late prophase stage. The intermediate of late prophase and anaphase stage, a metaphase stage, it will be perfectly observed. And here, last one is movement. Movement of all chromosome to the equatorial plates. They are going to the equatorial plates. Next one is metaphase. Metaphase means middle phase. Meta means middle. Formation. This is the formation of what? Formation of spindle fibers. Now the spindle fibers are completely visible. Alignment. The particular, just see into the particular metaphase stage here. This is the metaphase. Here all the chromosomes are situated within the metaphase plates at the remaining stage. So this is very important. Metaphase plates, what is it? And these particular spindle fibers are released through astral rays, are also released, but it is not visible in case of plants, astral rays, but the spindle apparatus is also available in case of plants. So spindle fiber attached perfectly with the centromeric region, that is called kinetochore. Okay, and it starts to pulse or contract. In the later we can see in the anaphase contraction of the chromosome to go to that specific poles. 50% of the chromosomes to the south poles, 50% to the north pole. So extra chromosomal fibers are also there. They are trying to pull through a stretches like extension, and after that separation. Separation means how these particular chromosomes are get separated from each other at the point of kinetochores. We can see here anaphase stage. Just see anaphase stage. This is separation. Separation of chromatids from each other for a single chromosomes. So 50% will be north pole and 50% will be south pole. This is an in-between stage where the movement is going on. Next one is separation is over. Next one is motion. Motion is means all the chromosomes, 50% chromosomes had going to reach to the north pole and 50% to the south poles, and spindle fiber helps to this movement. Telophase, we can see destination. All the chromosomes reach to the destination of the north pole and south poles. Reformation of what? Reformation of all the cell membranes, nuclear membranes and cell organelle membranes are getting visible. And evagination has been started. Here you can see the cleavage furrow-like structure. It is for animals, but for plants, invagination means cell plate-like structure has been formed in the center of the cells from here. We'll talk about later. Significance and differences. Mitosis. We just can see one particular animation, very short animations. Just see what we have studied here. We can see into this picture. Okay, so this is very basics. Now this is the cell cycles, interphase. This is mitosis, where karyokinesis, and this is cytokinesis. Just can see here, and from where the cell has been divided into two separate pieces. Okay, so these are the right marks means checkpoint. Next one is this is a cell where you can see that particular all the chromosomes getting condensed and is visible. Now they are starting to divide. You just can see centrioles are there. Centrioles are moved to opposite poles. This is for animal cells. Fully formed chromosomes are visible and nuclear membranes start to disappear. Now metaphase stage, the centrioles reach to that particular points. And just you can see here, cell metaphase plate has been formed here. All the chromosomes has been arranged at the centers. And after that, spindle fibers attached to the chromosomes at the chromosome centers, and they are trying to pull out all the chromosomes from particular this regions, spindle fibers, kinetochore regions. They are getting separated at the anaphase. Chromosomes get separated at the from the centromeres, and this is the anaphase structure. They are pulling one side, extra chromosomal pullings are there, and at the metaphase stage, complete nuclear membrane has been formed. All the chromosomes are getting decondensed and disappearing. Now we can see here telophase is complete. Now the particular invagination has been started to happen. Evagination has been started. So in case of plants, there are cell plates, that is called invaginations. This is due to deposition of cell wall, cell membranes. Okay, so from these pictures, as we can see, and we'll resume that our another part, mitosis in animals and plants. In prophase, centriole movements to two poles. So in prophase, one thing is happening, centriole movements. We can see here how in case of prophase, centriole is available in case of animals, but there is no centrioles. In case of plants, basically tubulin proteins are making that spindle apparatus. Which one? Tubulin protein. So many questions from this section. Tubulin. We just have to remember. So there is no such astral ray formation in case of plant cell. So in the next point, spindle apparatus. In case of animals, it is called spindle apparatus. In case of plants, it is spindle fibers has been formed, and it is in case of animal, it is with astral ray, and it is without astral ray. Cytokinesis at anaphase or telophase. That means in case of animal, the cytokinesis has been started at the earlier of anaphase. In case of plants, it is the beginning, at the beginning of telophase, the cytokinesis had been started. So the fourth one, invagination and invagination, as I talk about the cytokinesis completions process, cell plate formation, and later one, significance of mitosis. Now I have summarized in very short. What is it? Production, regeneration and replacement. Very short, you can remember. Production means number of cells are copied. Number of cells will be increased. Regeneration means what if the damaged cells has been formed due to external injury or any natural calamities. Plant cells repair to themselves through regeneration process. And last one is replacement. When the cells are getting aged at a particular point, the old cell has been replaced by new cells by mitotic cell division. So this is complete. Next video, next slides will be started for meiosis.